Hello, everyone. Good afternoon. Welcome to today's webinar organized by Cusabio. My name is Michael, and I'm from Cusabio Sales Department. And I will be the host today. We appreciate your time. On behalf of the company, I'd like to thank you for joining this webinar. In this webinar, we will discuss the background and technical details of single B cell technology. In addition, we will cover the specific features of single B platform from Cusabio and show you a specific case. As you see, single B cell antibody discovery technology is a powerful approach for isolating and characterizing antibodies from individual B cells. This method leverages microfluidics and high-throughput sequencing technologies to efficiently screen and clone antibodies with high specific and affinity. Compared with traditional hybridoma technology and phage display technology, the single B cell antibody technology boasts advantages such as a shorter development cycle, high throughput, numerous positive clones, and high affinity. Most importantly, antibodies obtained through this technique ensures the natural pairing of light and heavy chains. Today's webinar speaker is Dr. Ying Zhou, the product manager at Cusabio, where she oversees product marketing, development, and optimization. Dr. Zhou holds a PhD in biology and biotechnology from Wellcrest Polytechnic Institute in the US. After earning her degree, she completed postdoctoral training at the University of California, Davis. Dr. Zhou specializes in molecular biology and protein science with a focus on mRNA expression and regulation, mRNA stability, and RNA interacting proteins. The webinar will last for around an hour, including around 30 minutes presentation and 15 minutes discussion session. Please feel free to post any questions in the QA section, and we will answer them after this presentation. Now, it's time for Dr. Zhou. We're looking forward to an enlightening and inspiring session together. Thank you.
Can you hear me? Hello, everyone. Good afternoon. My name's Ying, so I'm the product manager from Cusabio. I'm so glad to see you again in this webinar. And today, we are going to talk about the single B cell technology, and we will see how it can accelerate the discovery of lead antibody molecules. So in the first part, I want to introduce the background of the antibody discovery platforms. We know that there are a lot of different discovery platforms, and also I will focus on the single B cell technology and introduce different types of the technology that you can see in the market. So let's start the journey today. This is the summary, or especially the timeline of the development of monoclonal antibody. So you see that the hybridoma technology kind of serves as a key to unlock the door to monoclonal antibody applications. And after that, we see the antibody phage display technology and also the single B antibody technology. So these new techniques applications felt the limitations of the hybridoma technology. So, but altogether, I will say that they made a great contribution to the monoclonal antibody development. So this is the summary of the platforms that for the monoclonal antibody discovery. And the first three we just mentioned, and then they are pretty common in the market. But the last one, the proteomics, it's kind of like a novel strategy to discover antibody, and I will introduce it in details later. First, the traditional hybridoma technology. So we see that after the immunization of the animals, especially with the designed antigens, we will fuse the B cells together with the melanoma cells, and the generation, especially the hybrid cells, they are kind of like generated. Then we can perform the sorting and the secreting to get the antibody secreting positive clones. Then we can perform the expansion of the positive clones. Then the cells can be transferred to the large-scale culture setups, and from there, we can produce the antibody in a large amount. The second platform is the antibody display technology. I will say that for this one, the key feature is that it can facilitate the selection of an antibody with the prescribed properties, especially that we are kind of like coupling the protein variant with the genetic information. So we can refer it as the genotype-phenotype coupling. So for example, what I showed here is one of the successful applied platform that in the antibody discovery, the phage display with the phage mid vectors. So you see that if we have different peptide that we want to display, or especially like the antibody candidates, we can fuse them and integrate it into the phage genome. Then after the expression, the antibody can be displayed on the phage surface. And with this different vectors, we can build a library of the antibody candidates with the desired characteristics. So it depends on what you want. Like, for example, if you want the antibody with the high affinity, like selectivity or stability, you can always design based on the genetic modulation. Then after that, we can kind of like do a screening for the library. So with this library of the different candidates, we can do the biopanning. You see that after 3 or 4 rounds the screening, the candidates proteins with the specific properties will be enriched. Then commonly we use the ELISA method to select it and detect it. So it's kind of like a pretty efficient way to get the antibodies that with the desired properties. And the third one is the one that I just mentioned. It's not that common. It's pretty novel. It's called the proteomics approach. And here is the overview of the proteomics approach to identify the functional monoclonal antibody. So basically, you also have the immunized animal. You can get the serum or plasma from the animal. Then after the affinity purification, the polyclonal functional antibody, they are purified. With this purified proteins, you can digest them. And finally, you will get a lot of like digested peptide fragments. Before the mass spec analysis, you need a reference database. So it means that when we do this, meanwhile, we need to have a reference database. And this reference database, they kind of like generated with the next generation sequencing. So you have to immunize the mice. Then you have the B cell source from the animals. Then you can use the next generation sequencing to build this reference database. So what we got from the previous digestion, we have a lot of peptide fragments there, right? Then with this database, you can perform the different analysis to identify the sequence. Then you can resynthesize and then do the cloning and the expression with like a matrix. You will get a matrix of heavy and light chains. With all this sequence, you can perform another in vitro functional validation to get the antibodies. So let's go back to the topic today, the single B cell technology. So we see single B cell technology, but they have a different types. So for example, here, the first type is based on the B cell sorting and isolation of the memory B cells. This one is usually that we rely on the phage to do multiple rounds of sorting to isolate the desired positive B cells. And the second one is a chamber-based culture. So this one, we focus on the plasma cells. With the plasma cells, we can culture them, and then we can perform the functional analysis to get the positive hits. The last one is pretty straightforward. It just like rely on the single cell sequencing. So direct the sequencing, you will get, okay. So let's talk about the single B cells technology. So the first one I just mentioned, this one is based on the B cell sorting and isolation. We rely on the fluorescent activated cell sorting technology. So basically, we have the mouse that immunized, and individual B cells, they are isolated based on the defined surface markers of the B cells. Then you use these surface markers to sort and isolate the desired positive hits, and the corresponding full-length heavy and light chain variable gene transcript that corresponding to this positive hits, they will be amplified. So you see, we have the heavy chain and the light chain that sequenced and amplified. Then up with the amplified fragment, we can cloning this amplified fragment into the expression vectors. So with this expression vectors, after the co-transfection into the host cells, we can produce the monoclonal antibodies in vitro. And then these monoclonal antibodies, they will have the antigen specificities that you want. So this is the first one based on the memory B cells, and then the strategy is based on the phage sorting and isolation. Then the next single B cell technology is based on the microfluid chamber based. So for this one, we focus on the plasma cells. I will say that for this platform, it's pretty convenient. It combines the cell trafficking, imaging, and culturing in the chip. So as I showed here, this is a single tiny chip. So inside this chip, there are some like a representative fluid structure. And with these different structures, we also have different chambers. In different chambers, we have the narrow scale volumes. So all this, it will provide a suitable microenvironment for the plasma cells to grow, and also at the same time, the spatial separation of these single cells, they are kind of like maintained. So each one is separated from the other one. And also inside this chip is kind of like a continuous perfusion culture system. This culture system makes the cells to grow robust. And then the numbers can be thousands of individual clones at the single chip at the same time. And inside this chip, additionally, the functional screening assays can be performed. The screening can be like a screened for the antigen specificity, cross-reactivity, and also like the function activity that you want to design. So then the positive clones after the culture and the functional screening, the functional screening, they will be identified and exported for the downstream process. So this one is focused on the plasma cells and then based on the culture chamber culture. The last single B technology is pretty direct. It just relies on the single cell sequencing. So with this, we just directly get the VH and the VL sequence of the antibodies from the individual B cells. So you can imagine that if you have thousands of B cells, we just directly sequence all of them to get the VH and the VL sequence. The whole system, I would say that is a pretty bioinformatics framework. With this framework, you can analyze the sequence data and identify the accurate VH and the VL pairing. So as you see here, we have different gel beads. With all these gel beads, the barcode information is inside. Then we mix the gel beads with our samples and also mix them with the enzymes. These enzymes will be very helpful for the downstream reverse transcriptase. So you see that after gel beads are dissolved, then the barcode sequence will be released, and the samples during the whole process actually they are labeled. So with the direct sequencing, you can quickly generate a very large database, and this database have the diverse panel of the antibody informations for each individual B cells. Then you do different analysis to get the good or excellent paired between the VH and the VL. Then you reconstitute the antibody in vitro. Then you will get the functional antibody. So from here is a summary slide. I want to focus on the advantage of the single B cell technology comparing to the other three antibody discovery platform. So if we compare the single B cell with the hybridoma, you see that because the single B cells the efficiency is high and it works in a high-throughput manner. So the development cycle of the antibody is pretty short. Then if you compare the single B cell with the antibody display, you will find that the single B cell, this platform can maintain the natural pairing between the heavy chain and the light chain. And also because we will get the vector co-transfected into the host cells. And with the mammalian host cells, the post-translational modification profile of the antibody is maintained and is native. And also with the single B cells, because we can start from the immunized mice. So the diversity of the antibody is broad, and we can find a lot of diverse antibodies with this one. And comparing to the last, the proteomics, because everyone knows that the next generation sequencing is pretty expensive. So comparing to this one, the single B cells have a low cost, and the manipulation is quite easy, and also the accuracy is high and guaranteed. So we see a lot of advantage of the single B cell technology. Then with all this knowledge, I want to move to the second part. So what about the Cusabio single B platform? What are the features from the Cusabio? And after that, I want to present our internal case from Cusabio, and then show you what we got and how the data looks like. So the first Cusabio feature I want to mention, the first one is that we perform 2-round screening. So the overall method of Cusabio single B platform is based on the phage screening approach, but different from others, we perform 2-round screening instead. We targeting both the plasma cells, but also we targeting the memory B cells. So how can we do this? This is really because that if we think about the differentiation from memory B cells to the plasma cells, the differentiation, there's a very short transitional stage between this differentiation stage. So which means that there's a very, very short time window between the memory B cells differentiate to the plasma cells. So we have a special technique that we can perform the 2-round screening relies on this very short time window. So that reason we can target plasma cells, we can also target the memory B cells. And with all this, the sorting efficiency of the antigen-specific B cells, the frequency increased, and then definitely the success rate of our project is also increased. This is our first feature. Then what about the next one? The next feature we introduced into our platform is the construction of linear expression vectors. So comparing to the traditional method, we see that after the antibody the gene information was amplified, usually the amplified gene, they are ligated to the digestive plasmid. Then you begin from here to get the clones, you scale up the culture, and then you will perform the plasmid extraction again. And then you perform the co-transfection into the host cells. For the Cusabio platform, actually, we do not have the plasmid construction stage. So we save 3 days. Instead, after we got the gene amplified, we ligate it to the regulatory elements. So these regulatory elements include the operate promoters, terminators, and some other regulatory elements that's necessary for the expression. We call it transcriptional active PCR. So we skip the plasmid construction. We directly perform the co-transfection. So 3 days saved. That reason with our platform, the efficiency increased a lot. And also, in addition to that, because we do not need to, construct a lot of different plasmids, so the cost of our platform, the technique, the cost lower down. And we also guarantee that even though the efficiency increased, the cost lower down, we still have the microscale supernatant. We can guarantee that the microscale supernatant can meet the basic needs to do some functional detection. So for example, like the ELISA detection and the phage detection. So with all these 2 features, here let's take a look at the Cusabio single B cell discovery pipeline. So this is the whole pipeline for each project. First, we perform the animal immunization. Then we will correct the spinning lymphocytes, isolate them, and prepare them. And with our featured sorting strategy, we perform 2 rounds sorting to enrich the single cells, the positive cells, and we save the time in the plasmid construction. We directly construct the linear expression cassettes to get the microscale culture. And all this microscale culture, they are sufficient to perform the functional screening. Then after the initial functional screening here, we scale up the culture again, and then we perform the plasmid here. Then we have the large amount of the antibody again to perform the second validation. So this is the whole pipelines that in our project. I know the data is always, everyone cares. So here is the internal case presentation. So I would say this one is just to show to our customers. So I will say some limitations for this internal case. The drug target that we start with, this one is TRPR2. TRPR2 is a pretty popular and hot target in the ADC. So ADC is antibody drug conjugates. This one is a pretty novel and efficient therapy in the market. You can see that for the ADC, especially targeting the TRPR2, we already have the approved ADC in the market, and we have a lot of different clinical trials from different phases targeting the TRPR2 in the, they are under the clinical trial, and then hopefully we will see them a lot approved in the future. At the very beginning, we usually, we prepare the antigen of ourselves. So this is the SDS phage showing that we successfully expressed the full-length TRPR2. And this is the ELISA binding experiment to show that the TRPR2 target is specifically binding to the positive antibodies. And then this is the animal immunization. This chart here summarizes the immunization mice we have and the immunization timeline and also the immunization method we used. So to be clear, we have 2 different species of mice. We prepare the BAPC and the AJ species, and for each one, we have the two mice for each species. This is the 4 different immunization we did. And after the 3rd immunization and then the 4th immunization, we will perform the serum titer testing to confirm the immunization result. This is the ELISA experiment that we did to confirm our immunization result. As you see, here is our negative control. This data come from the pre-immune serum data samples, and this is the result from after the 3rd immunization, and this is the 4th immunization. This is the chart showing the ELISA data. As you see, it looks like that the AJ species, the mice, they have an excellent behavior comparing to the BAPC. So we continue with the AJ, and then we did another serum titer testing with the phage. So the previous one is ELISA, and this one is the phage. The phage flow cytometry is worked with the endogenous cell line, the MCF7. So to see whether there's a specific binding like at the cell level. And BMK is our control. So we got some the positive antibody from the market to work as a control. So BMK is always the positive control. And as you see, the AJR4, this mice, looks like it has the best result comparing to all the other mice and all the other immunization group. So we continue with the AJR4, and we move with this one to correct the spinosite. So you see, this is the flow cytometry that we performed to start and screening the B cells. Altogether, there are 6 different screening and sorting. We expect that after the final screen, we will get more than 3,000 target cells to get to go with the next stage. But as I said before, this case is an internal case. So instead of with like a large number of the cells, we choose to start with a very small number. So inside this more than 3,000 cells, definitely, actually we choose the 48 cells to sort into the individual well. And we mix the 48 cells with the reverse transcriptase mix. And then we chill to 4 degrees in the 96 well PCR plate to generate the CDA. So for all these 48 cells, the CDA are generated. Then we perform the reverse transcriptase PCR to get the heavy chain and the light chain variable region, the gene amplified. So as you see here, the left is the amplification of the heavy chain, and then this is the light chain. For the amplification, you see that all the heavy chain, the gene are successfully amplified, but for the light chain, only 44 are amplified. But then we move on with this 44 different clones. So this is the right here is a construction of the linear expression cassette. The left is heavy chain and the light chain. So all 44 clones, the heavy chain and the light chain, they kind of like successful constructed. So I would say that these two, they are separate linear transcriptional activate PCR products. So heavy chain individually and the light chain individually. So after the cassette construction, we performed the post-transfection of this linear cassette into the host cells. Then after the recombinant antibody expression, we performed the initial functional validation with ELISA and the FACS. So this whole table is the raw data for our ELISA and the FACS. So the green is ELISA and the FACS is the orange is the FACS. So you see all 44 positive clones from the previous, they are here. And you see the result here. So in summary, for the ELISA, we got 34 positive hits. So the positive rate here is 77. And then after the FACS, we got 24 positive hits, and the positive rate right now is 70%. So if you think about our initial material, we got 48 at the very beginning. And after the FACS validation, we finally get 24. So I would say that the positive rate overall for this internal case project is 50%. So we got a lot of positive hits, but I know that most of the customers, they not only cares the number, but also cares about the diversity of the antibody. So we also access the diversity with the CDR3 sequence. So for the positive clones, the previous 24, we perform we extract the CDR3 sequence, and we use the sequence to access the diversity. This is because that the CDR3 is always related to the specificity and affinity of the antibody. And I know that in the published papers, they always perform the clustering analysis to determine the diversity. I would say that if the CDR3 regions, the identity is higher than 80%, they are usually well grouped to the same family. But here, as you see, our identity is definitely less than 80%. For the VH, the identity is 47, and for the light chain, the identity is 66%. So both of them, they are lower than 80%. Then from here, from the diversity assessment result here, I will say that not only like our positive rate is high, but also our antibody diversity is also guaranteed. The last part for this internal case is that we also choose the top 3 unique sequence from the previous batch ranking result. And we express these 3 sequence and we validate them again with the ELISA and the phage. So in this second validation, we choose the positive antibody that we got from the market to work as a positive control. And we first compare our 3 clones with the positive group. You see that the result is pretty consistent, means that there's a specific binding of these antibodies with the antigen. And then the next, we move to the flow cytometry experiment to detect the ability of these antibodies to bind to the cell surface antigens. And I'm so proud to say that our clone number 2 and clone 24, they have a very good performance. Their performance is even better than the positive group. You see that it's definitely higher than the control that we found in the market. So make a conclusion here. So in our internal case with the single B antibody discovery, we have a very high positive rate and our antibody diversity is guaranteed. And in the final validation, we find that our top unique sequence have a very good performance, even higher than the positive group. So at the end, I want to make another a final summary about the timeline of our single B cell antibody discovery. Excluding the animal immunization, at the day 1, we will correct the mouse phenotype, and then we will perform the single cell sorting and isolation based on the facts. Then from day 2 and day 4, we will amplify the VH and VL and perform the construction of the linear cassette, and then directly go to the co-transfection to get the microscale supernatant for the validation. So you see that at the day 8, we already have enough supernatant to perform the functional analysis. Then after another 14 days, we will get the milligram scale purified antibodies in a large amount to deliver to our customers. So in one sentence, our functional antibodies in small scale supernatant are available as little as 8 days. And if our customer wants the milligram scale purified antibodies, it will be obtained within the 22 days. So you see that with the single B cell antibody discovery platforms, the timeline can be pretty short. So at the end, please give me several minutes to introduce our company. So we are Cusabio. Cusabio represents that we want to make our customer satisfied biology. And our product line is pretty broad. We have the ELISA kits, and we also have the proteins, antibodies. And in addition to that, we also have the molecular biology product and the small molecular antigen. Our products has been exported to a lot of countries like the Canada, the USA, and then Russia, and some Australian. And also the cooperation between us and the big companies, they are pretty lot. Like we have cooperation with Pfizer, Sanofi, and Bayer, and Zinker, Johnson & Johnson. So you see that we have a very good relationship with all this. Then thank you so much. And I will say that if you want to stay connected with us, please scan this barcode and follow us in the LinkedIn. Thank you so much. And I will take the questions.